have the lecture number 28, which are the last 10 slides of the joint lecture number 27 and 28, because the file size was too much, therefore I have to divide it in two parts. So this slide describes about the role of the hormones epinephrine and glucagon on glycogen. So both of these hormones break down the hormone glycogen, and their mechanism of action is shown. A correlation is there from the lectures that we took on biosignaling, as at least epinephrine hormone was there in quite a bit of detail. So you must have some background knowledge of how these hormones work. So first up, the hormone binds to its specific receptors that are on the cell, epinephrine on the myocyte and glucagon on the hepatocyte. So this is their primary organs of action. Epinephrine mainly works on the muscle cell and glucagon mainly works on the hepatocyte. When these hormones bind to their receptors, both of these receptors are G protein coupled receptors that we discussed in detail in biosignaling. So this is going to activate the G alpha part, which is bounded with a GTP molecule. So G alpha becomes active, detaches from the rest of the G protein and activates the enzyme adenylyl cyclase. And this enzyme converts ATP to cyclic AMP, and cyclic AMP is the second messenger for this pathway basically. So CAMP or cyclic AMP in turn is going to activate the protein kinase A or pKa, which in turn activates phosphorylase B enzyme or phosphorylase B kinase enzyme, which eventually activates the glycogen phosphorylase, which is the main enzyme of the glycogenolysis pathway, or the pathway in which we break down glycogen we discussed last time, and glycogen phosphorylase is the main enzyme. So this is a cascade of reaction, as we call it. One reaction gives or activates another reaction, and the product again activates another reaction. And our main concern, or our main enzyme, you can say, is glycogen phosphorylase. That is the actual enzyme of the glycogenolysis pathway. So this is what we are trying to activate, and it is done so with the help of these two hormones, epinephrine and glucagon. And this diagram is also showing us the amplification of the signal, because initially we have started off with one single molecule of epinephrine and glucagon, and the end result is around 10,000 glucose molecules getting released from the hepatocyte, and almost the same number that will be oxidized in the muscle tissue as well. So starting from 1, we get around 10,000 X molecules, or the glucose molecules, which is a great increase in number. Therefore, this is what we call amplification of the signal. Here we have a little bit of detail about this enzyme glycogen phosphorylase individually in the muscles and then in the hepatocyte. So the control of glycogen breakdown is mainly via these two hormones, glucagon and epinephrine, as we saw in the previous slide as well, and their signaling pathway. We discussed the diagram previously. When the hormones activate the pathway, basically it is going to be through the second messenger system, CAMP or CMP being the second messenger. Eventually glycogen phosphorylase gets activated, which is the main enzyme of glycogenolysis, and this enzyme starts to break down glycogen. Now this, how does it do that? Again, we discussed this glycogenolysis pathway in detail. It's going to break one glucose molecule at a time, and the product is glucose 1-phosphate. Later on, we converted this to glucose 6-phosphate with the help of phosphoglucomutase enzyme, but that point is not mentioned here. So let's stick to the product glucose 1-phosphate, which is going to be broken off from glycogen one by one by the glycogen phosphorylase enzyme. Now the point to note here basically is that this glycogen phosphorylase can exist in two forms which are interconvertible, and one of the forms is active and one is inactive. So the thing to remember is which form is active. It is the phosphorylated form, the one that has got these phosphates attached with it. As we can see at the bottom, this is the phosphorylase, which is active form when phosphates attach with it. And if we remove these phosphates, it becomes the inactive or the less active form. So dephosphorylated form is inactive, phosphorylated form is active. And this phosphorylation and dephosphorylation, I think we have discussed at least one or two times earlier as well. I told you earlier that the kinase is always the enzyme that phosphorylates. So this one is going to make it active, whereas phosphatase is the enzyme that breaks the phosphate away or dephosphorylates some molecule, so it is going to make it inactive. So glucagon in the liver and epinephrine in the muscle, along with these other molecules, calcium and AMP, all have a positive effect on this kinase. Therefore, the phosphorylase becomes active under the influence of these molecules. And the active form is in the muscles. As we are talking about the muscle phosphorylase, it is going to be active during the vigorous exercise or vigorous muscular activity, whereas when you are resting or not doing anything, then it will be the inactive form that exists more. Now, apart from the glucagon and epinephrine, the hormones that activate this enzyme, glycogen phosphorylase, there are two allosteric control mechanisms as well, which I told you in the last slide as well. Calcium ions and AMP. Both of these molecules also activate the glycogen phosphorylase through the kinase enzyme. Basically, calcium binds to the kinase and promotes its conversion to the active form, which is phosphorylase A, whereas AMP accumulates in the vigorously contracting muscles when the ATP gets broken down, because your muscles are using plenty of ATPs during exercise. This one also binds and activates the phosphorylase enzyme as well. When ATP levels will be adequate again, then this activation will be shut down or stopped for some time. In a nutshell, simply remember that these two molecules, that is calcium ions, Ca plus 2 and AMP molecule, these are what we call the allosteric activators of glycogen phosphorylase enzyme. So these two are activating the enzyme allosterically. Next up is the same enzyme, glycogen phosphorylase, in the liver. So in the liver, main thing to remember is the main hormone that acts on this enzyme or controls its activity is going to be glucagon this time, not epinephrine. Two forms of this enzyme exist similar to the muscle phosphorylase. Dephosphorylated form is inactive, phosphorylated form is active. So up till here it's exactly the same as the muscle phosphorylase. This one is also hormonally and allosterically regulated, so this is also a same property like the muscle phosphorylase. Now when the blood glucose is too low, glucagon will be the hormone that acts through the cascade mechanism that we saw earlier, one reaction activating the next one and so on, and eventually converts the phosphorylase B to its active form A. So glucagon activates this glycogen phosphorylase in the liver with the help of the signaling pathway cascade. Now when the glucose goes back to normal, glucose enters the hepatocyte and binds to the inhibitory allosteric site on phosphorylase A and therefore inactivates it. So the molecule that is allosterically going to inhibit or stop this molecule is this time glucose itself. So do remember, glucose is the allosteric inactivator this time of this molecule phosphorylase A in the liver, whereas in the muscles we had two allosteric activators, calcium and AMP. Here we have a allosteric inhibitor, which is glucose itself. So this liver glycogen, guys, is liver glycogen phosphorylase is going to act as a sensor of glucose molecules. As we saw in the previous slide, for this enzyme, glucose is the allosteric inhibitor. So glucose, actually this enzyme phosphorylase acts as a sensor of glucose molecules in the blood. Whenever glucose levels are too high, what happens is glucose binds to one of the allosteric sites of this enzyme phosphorylase in the liver, and it causes a conformational change that exposes its phosphorylated serine residues to the action of phosphorylase A, phosphatase, or PP1. And this PP1 basically converts the phosphorylase to the B form and sharply reducing the activity. In simple words, it is going to inactivate the enzyme. So glucose is the allosteric inhibitor, which causes the inactivation of phosphorylase A when its concentrations are too high, and this is done so indirectly, you can say, with the help of the phosphatase enzyme. And what the phosphatase does is removes the phosphate from it. As in the diagram below, we can see this is the phosphorylase enzyme that breaks glycogen, and it has got the two P's phosphates attached with it. Therefore, it is the active form. When the glucose concentrations are too high, it is going to act through this phosphatase enzyme and removes the phosphate from it. As we can see, the two PIs going out. Therefore, this phosphorylase loses its phosphates and becomes dephosphorylated, which is the less active form. So insulin basically indirectly stimulates the PP1 and slow down the glycogen breakdown, and it also inhibits this phosphorylase enzyme. Again, the same effect stops the breakdown of glycogen. So as we saw in the previous slide, on one hand, insulin has an indirect control of the glycogen breakdown, where it stops the breakdown of glycogen. And on this slide, we can see that insulin has got a direct control over the synthesis of glycogen this time. So whenever the glucose levels are high, this will be a signal for release of insulin, and insulin signaling pathway gets activated. Now this insulin has got three effects or three main functions to perform. First up is the increased glucose uptake, or we can call it increased glucose import into the muscle. So more and more glucose will be taken inside the cells, which is shown here as myocyte. It will stimulate the activity of the enzyme hexokinase, which is one of the main enzymes of glycolysis. So in this word, you can say it is activating glycolysis, or in other words, it is trapping the glucose inside the cell. This free glucose can go outside as well, but once this hexokinase attaches a phosphate with it, now this glucose 6 phosphate has to stay inside the cell. It does not have the ability to leave the cell. So this reaction is mainly for trapping of glucose. And the third effect is the activation of the main enzyme of glycogen synthesis, which is glycogen synthase. So glucose 6 phosphate becomes glucose G1P to GDP glucose, and later on it's going to form the glycogen molecules with the help of this enzyme. So insulin has a positive effect on this molecule, as we can see, this enzyme glycogen synthase. So the overall effect will be to take more and more glucose inside these cells and store these extra glucoses in the form of glycogen. Therefore, when you are not eating, we can break down this glycogen and use these molecules for energy purposes. So that is the direct control of insulin over glycogen synthesis. In a nutshell, it does these three, you can say, functions and increase the synthesis of glycogen molecule because there are plenty of extra glucose available at this time. So just like glycogen phosphorylase that existed in two forms, this enzyme as well has a similar mechanism. Glycogen synthase also exists in two forms. So one is phosphorylated, one is dephosphorylated. But its phosphorylated and dephosphorylated forms are opposite to glycogen phosphorylase in terms of activation and inactivation. And the difference is here the phosphorylated form. Here we can see the P's with the glycogen synthase. So the B form or the phosphorylated form is inactive here, whereas the glycogen phosphorylase that was active when the phosphates were attached. Glycogen synthase is inactive when phosphorylated. So to make it active, we will have to remove these phosphates. As we can see on the right side, glycogen synthase A is the active form which does not have phosphates attached with it. So dephosphorylated form is the active form, whereas whereas the phosphorylated form is the inactive form. That is one thing that you must remember out of this slide. So phosphorylated is the inactive form. Now how to make it active or inactive, we have got different mechanisms. All of these green colored signals at the bottom that you can see, these molecules, insulin, G6P or glucose 6 phosphate, and glucose itself, all have a positive effect on this reaction going from left to right side. So these molecules are making the glycogen synthase active, and it makes sense as well. Insulin has a positive effect on glycogen synthase, also glucose and glucose 6 phosphate, because these are the actual molecules that are going to be used in the synthesis of glycogen. Whereas insulin has an indirect effect on stopping this GSK3 enzyme, and what this enzyme does is makes it inactive. So if insulin stops this enzyme, it means that the already active form cannot become inactive. So in a way you can say this is doubling the effect of insulin. It's catalyzing or it's activating this reaction catalyzed by PP1, so makes glycogen synthase active, and also indirectly prevents it from becoming inactive by inhibiting this GSK3 enzyme. And this effect is written here. Glycogen synthase is phosphorylated on various residues by this GSK3 enzyme, glycogen synthase kinase 3. So this is the enzyme that makes it inactive, and insulin can also inhibit this GSK3 enzyme so that glycogen synthase does not become active. Also remember, opposite is the effect of glucagon and epinephrine hormones, which are stopping this PP1 enzyme, therefore stopping the glycogen synthase from activation. So when we have glucagon and epinephrine more in circulation, that would mean that glucose levels are low in the body. So we want to stop this glycogen synthesis pathway, and that is exactly what is being done here. Here we have the indirect effect of insulin on glycogen synthase through this enzyme GSK3, and this I described in the previous slide as well. Insulin has its enzyme linked receptor, insulin receptor. No need to go into too much details, but what is the overall effect is what is important. When the insulin binds with its receptors, goes through the cascade of events. It works through the P2 second messenger system, and the overall effect is to make this GSK3 inactive. As we can see here, it's going to activate the PKB enzyme, which converts GSK3 into the inactive form. Now once this GSK3 is in the inactive form, that is what the main purpose of insulin is, basically to inactivate this GSK3. Once it has been inactive, the function of GSK3 was to convert the glycogen synthase into this inactive form. So GSK3 is inactivating the glycogen synthase. Now if we can stop this GSK3 from functioning with the help of insulin, it means that the glycogen synthase will remain in the active form, and that is the whole purpose. Basically, we want this glycogen synthase to be active under the influence of insulin. GSK3 is making it inactive, so we stop the GSK3 with the help of insulin by making itself inactive, as we can see here. Again, the mechanism is phosphorylation and dephosphorylation. Don't really need to go into too much detail, but this phosphorylated form of GSK3, which we can see with the phosphate, is the inactive form. So this is insulin's activation of glycogen synthase, you can say indirect activation by inhibiting the GSK3 enzyme. Next up, we have the control of carbohydrate metabolism in the liver organ, liver mainly, and that is done with the help of the two hormones that we have already discussed. Most of these points we have already touched one way or the other, basically. So it's kind of a summary of this carbohydrate metabolism in the liver under the influence of the two hormones, that is insulin and glucagon. Let's start from the top with the insulin. So insulin is released when the blood glucose levels are high. That is a signal. So we have high concentration of insulin, and under the influence of high insulin, we also get high amounts of GLUT2 transporters, and that means more glucose will be taken inside the cell. That is increased glucose uptake. Insulin itself is going to work through these cascade of reactions, which you don't really need to memorize each and every one. But what you need to know is insulin is going to stop or inhibit glycogen phosphorylase. As we can see, the little arrow pointing downwards means its activity is decreased by insulin. If you want to go into detail, then this is done by the insulin sensitive kinase, increasing the activity of PP1 and decreasing activity of phosphorylase kinase, and these things were there in the diagrams, the cascade reactions in between. But even this much is enough. In high levels of insulin inhibits the glycogen phosphorylase. Also, high levels of insulin activates glycogen synthase, as the arrow is pointed up, and this is done through this GSK3 enzyme inhibiting that we just saw in the last slide, which is in turn done by the PKB enzyme. Again, some unnecessary details, but at least for this one, do remember this GSK3 inhibition, because that the whole slide is about this enzyme as well. So in a nutshell, insulin is decreasing glycogen phosphorylase. This enzyme does what? It breaks down glycogen. Therefore, glycogen breakdown is going to be decreased, as mentioned here, and glycogen synthase synthesizes glycogen. Therefore, glycogen synthesis will be increased under the influence of insulin. Taking more glucose inside the cell means we can use more glucose in the cell. Therefore, glycolysis is increased through insulin. So these are the three main, you can say, pathways of glucose or carbohydrate in the liver. Two of them are activated, glycolysis and glycogenesis are activated by insulin, whereas glycogenolysis is inhibited by this insulin. Exactly opposite thing is going to happen with the glucagon. So here we have the low blood glucose levels this time, and those low blood glucose levels are a signal to activate glucagon or secrete glucagon. It works through the CMP system. So CMP is its second messenger, which activates the protein kinase A, and it's going to have its effect on the same three metabolic pathways in an opposite way. So glucagon increases glycogen phosphorylase activation, which is going to increase the glycogen breakdown, and that is what we want. When we have low blood glucose levels, we want to produce more glucose. If we break down glycogen, we get more glucose. Glycogen synthesis will be decreased by this enzyme, glycogen synthase, so it becomes inhibited, and we decrease the glycogen synthesis. Therefore, we don't use too much glucose for synthesizing glycogen. We are actually breaking glycogen because we need glucose. And also through this main enzymes of glycolysis and gluconeogenesis, inhibition of PFK1 and activation of FPPase2, we decrease glycolysis, and that is also our purpose, because we want to use glucose very carefully now. We don't want to waste it. So these are the three effects of glucagon on the carbohydrate metabolism, decreasing glycogen synthesis, decreasing glycolysis, and increasing glycogen breakdown, that is glycogenolysis. Next up, we have a repetition of a slide. Maybe some additional text is here, which was not there in the previous one, but it's exactly the same. The control of the carbohydrate metabolism in the liver versus the muscle during the exercise, and the hormones that are shown here are glucagon and epinephrine. Both have similar function to play. I've already told you. In the liver, both have exactly the same effects basically. Glucagon mainly breaks down glycogen, and that produces glucose 6 phosphate, which can go out into the blood glucose because we can free the glucose. Liver has the enzyme phosphatase that can remove this phosphate and push this glucose into the bloodstream. Pyruvate can be converted into glucose 6 phosphate as well through the process gluconeogenesis. So gluconeogenesis, we can see an increase under the effect of glucagon, and glycogen breakdown or increase in glycogenolysis we can see as well. Both epinephrine and glucagon are doing the same thing in the liver. But the main difference was that in the liver, glycolysis is being decreased, as we can see the arrow downwards, because liver does not need plenty of energy at this time. Instead it is going to throw its glucose into the bloodstream where vital organs can use it, like brain or kidneys. Glucose is more important for them in this situation of starving. But for the muscles, because the muscle cells do not have the enzyme glucose 6 phosphatase, which can take this glucose out into the bloodstream. As we can see here, this can go out because of the phosphatase enzyme, but this glucose cannot go out into the blood because the muscles don't have the glucose 6 phosphatase enzyme. Therefore, this glucose obviously is going to be used within the muscles. Rest of the effects are the same. Glycogenolysis will increase, so we break the glycogen into glucose, but instead of gluconeogenesis, which can produce glucose from pyruvate, there is no effect on gluconeogenesis here. Instead, glucose 6 phosphate will go down the pyruvate, which is glycolysis. So increase in glycolysis is shown here because that is what we want. Muscles in time of this starvation or under the influence of epinephrine, actually it's going to be an emergency situation where you need your muscles, and if you want to use them, obviously you will have to get some energy for them as well. Energy will be provided through this glycolysis pathway by breaking this glucose down into pyruvate, whether oxygen is available or not available. So no gluconeogenesis in the muscle, that is one thing to remember. And increase in glycolysis, active glycolysis during vigorous contraction will be seen in the muscle but not in the liver. And active glycogenolysis, obviously breaking down more glycogen so that we can get more glucose. So in the text is simply on the skeletal muscles effect that is shown here.